So gastrulation, as I mentioned to you, is a critical part in our lives. So our development comes from a single cell, and even for the queen, from a single cell. The cells divide and at first make a hollow, simple ball of similar-looking cells called the blastula, shown here. And then, but we need to generate all of the rest of the organs of our bodies here, and this happens first through the process of gastrulation, when part of the embryo is pushed inside this blastula to first give rise to 2 layers of cells, which eventually give rise to 3 layers, as shown here. So this is a typical representation of a gastrula, in which you have the external layer called the ectoderm that will give rise to skin and the nervous system. Inside, we have the endoderm that gives rise to the gut, liver, and everything sort of in our digestive tract. And in between, we have the mesoderm that will become muscle, blood, the heart, and other important internal organs. And so it's an important step in an animal's life because we don't want to go around with any of these important systems. And so gastrulation is a really beautiful process. And it has inspired David and all of us to create the cellular dance. Look at this. This is a gastrulation in a sea urchin embryo. And each yellow spot here marks an individual cell. You can see how they are brought inside and invaginate from the outside of the embryo inside to form these new internal structures. But let's go back to our favorite organism, the fruit fly. Gastrulation in Drosophila has been studied for a while, and we now know a great deal, but not quite everything, about the processes that occur during gastrulation. This is a Drosophila embryo. The ventral side is shown here, and in this micrograph, it is caught in the middle of the process of gastrulation. So you can see that these cells on the ventral side of the embryo are moving inside. This happens through the process which is called apical constriction. Originally, earlier on in development, the apical sides of these cells have this dome-like shape. Right before gastrulation happens, these cells first flatten, and then they constrict at the apical side. This means that this side becomes shorter while the bottom of the cells is still wide. So they, in cross-section, they become trapezoidal in shape. Because this happens to a lot of cells simultaneously that are touching each other, the whole layer of cells is pushed inside. It invaginates inside. Now we know a great deal about the molecules driving this. These are our friends, actin and myosin. A lot of this work has been done in the lab of Adam Martin, where they developed an interesting model of a so-called radial sarcomere. I showed you the structure of a linear sarcomere which is present in our muscles, but here we have a radial arrangement of these molecules. So actin filaments are arranged around from the center of the cell outward like this. And then myosin forms a tight ball in the middle, in the center of the apical region. And when it's time for them to contract, then myosin tries to crawl to the plus end of the actin molecule embedded in the periphery here, and that causes apical constriction and the narrowing of the top of the cell. It is nicely shown in this video. This is the apical side of these cells that coordinately constricts and becomes narrower and narrower while the bottom of the cell is still wide. This causes the whole group of these cells to coordinately move inside and form a structure which is called the ventral furrow in the embryo. And so you may have guessed, gastrulation has to start with a signal. What is this signal? Through the work of many labs, we now have a pretty good idea about the signaling pathway that sets up the process of gastrulation. It starts with a signaling molecule, extracellular signal, that is called FOG, that stands for folded gastrulation. The rest of the pathway goes through several steps of signal transduction to finally arrive and at the myosin molecules that get activated in the process and cause apical constriction. Now notice that the fog is interacting with a receptor on the surface called mist. The receptor for the pathway was elusive for many years, despite the fact that the extracellular molecule and some other molecules inside have been identified. And notice how creative fly people are with naming our favorite molecules. Look, folded gastrulation is abbreviated FOG. What is the most appropriate name for a receptor? Mist. There is a French group that identified another receptor, which is also helping fog to signal. They ran out of good names, and so they named their receptor Smog. I'm not joking, you can look it up. Yeah, Smog is a real protein. But anyway, so it acts in a way similar to mist, and finally we have our myosin activation. These molecules are placed in the correct region of the embryo, which is the ventral side, at the correct time by a whole network of other regulators that set up proper positioning of these molecules at the right time to form the ventral furrow. Now, an important question here is seemingly we have all the parts that are necessary for this. But it's important to remember that signaling pathways have to be reset to their original state. Think about our visual perception of light. If the visual system was not reset into an original resting state, we would be continuously overcome with light and blinded because we would not be able to perceive more light photons. So it is important to set up certain blocks to excessive signaling. And this is illustrated in this diagram. Here, we're looking at the ventral side of embryos. This is a control fly embryo that has successfully completed gastrulation, and you can see it forms a nice ventral midline shown here. This is an electron micrograph of an embryo that has excessive fog. You can see that things are not very good looking here. The cells have not completed gastrulation properly. They're lingering on the outside, and the ventral furrow cannot properly form. For a number of years, my lab has been working with a signal controlling protein called KERTS. And when we looked at KERTS mutant embryos, we couldn't help but notice a similarity with the embryos that have excessive fog. Now, I have to tell you what KERTS is. KERTS is a protein in Drosophila, which is very similar to human proteins called beta-arrestins. They are called this way because they stop or arrest the signaling from a class of receptors in mammals and in flies called GPCRs. And they do this by internalizing the receptor away from the surface of the cell inside so that they no longer can interact with extracellular signal molecules. And so we suspected that KERTS may play a similar role in Drosophila, and MIST, being a G protein coupled receptor, was a perfect candidate for KERTS to be repressing. And so through a lot of experiments that I don't have time to show you, I will show you a few. We now know that pretty well that this is how it happens. So KERTS will directly associate with the MIST receptor and prevent excessive activation through fog. A lot of this work is done by a graduate student in the lab, Faye, who is here today, and he will be showing you some embryos in that fluorescence microscope. So let me show you a couple of videos that show you what happens in the embryo that doesn't have KERTS. First, let's look at the top here. This is the normal embryo, and when the process of gastrulation happens, you can see a spike of activation of myosin shown in green here, and its concentration in the middle of a cell. Then the cells can relax after that and internalize into the embryo and complete gastrulation. Look what happens in a mutant embryo that has no KERTS. First of all, the range of cells, the width of cells that try to carry out apical constriction is much wider. The cells contract randomly, and they cannot get out of this contractility. And so the embryo, unfortunately, even loses the cellular structures and essentially tears itself apart through the process of random constrictions. So it's important to have Kurtz around to turn the signaling off. Now, lately, we have been looking at the structure of Kurtz. This is a molecular representation of a Kurtz molecule, which is based on the known structure of a human protein to which it is very similar. So Fay has been trying to map the important regions in this molecule to see which ones are important in development. And so we recently focused our attention on a region which is called the finger loop here, shown as these yellow balls here. And so it is called this way because it is thought that this region directly interacts and inserts itself into the G protein coupled receptors. And so just to illustrate, to give you one example of this result, look at what happens if you introduce just this one mutation into Kurtz. On the left here, you see a control embryo at the embryo stage right after gastrulation. And eventually, it will form a nice-looking larva, shown here. They look like little worms and crawl around. And Melissa, during the intermission, will show you some fluorescent larvae here. Without Kurtz, this is a mild phenotype of an embryo that does not have Kurtz. It couldn't complete gastrulation, and unfortunately, at the larval stage, they do not survive, and instead they get a big hole still open on their ventral side. When we introduce a mutation of just these two amino acids into the finger loop of Kurtz, we get similar looking phenotypes. The embryos cannot complete gastrulation properly and end up with an opening like that. So as you can see, it is as important to activate the pathway as it is to turn it off eventually. And so this is our summary model of what we think is happening. During normal signaling, FOG will properly activate the MIST receptor to cause normal apical constriction. Eventually, this process has to be terminated by the activity of Kurtz, which will directly interact with the receptor and dampen and cancel any further constrictions. If you have a mutant molecule of Kurtz, it cannot properly associate with the receptor. And so it causes excessive apical constriction, which is extreme in the embryos that have no Kurtz at all. And so I would like to wrap up my presentation by bringing parallels between Drosophila and humans. So there are a lot of processes in our development that are based on cellular contractility and controlled cell movements. So for example, this is a neural tube closure event in the mouse embryo, and it looks very similar in the human embryo as well. And a lot of human diseases are associated with abnormal cell movements, such as spina bifida when this process doesn't complete properly, or excessive migration of cancer cells in the body. And our humble hope here is that by using flies as a model system to try to understand these processes, we can, first of all, help understand our development better, and perhaps maybe even find therapies for some human diseases. So to summarize the third part of the talk, gastrulation is important because it will establish all of the future organ systems in the organism. It involves actin-myosin contractility and concerted cell movements that we all like to call cellular dance. And so gastrulation is initiated by signals, and it is very critical to keep the signaling just at the right level. So mutations that disrupt both the generation and the stopping of these signals are detrimental for development. And finally, we're all hoping that flies will eventually help us to find therapies for human diseases. Okay, this is the end of part 3 of my presentation.